Okay, so now we're going to start talking about just basics of biochemistry. And so the first few chapters of Mark's biochemistry, medical biochemistry, are involved in just very general overviews of fuel metabolism. So, what are the fuels? What does your body really need? What is the difference between fed and fasting states? What does that mean at the level of biochemistry? And so we're going to start by a very general overview of the biochemical processes in your body. This figure here basically takes you through everything that we are going to be covering in Biochemistry 1 and 2. And it all starts with our diet, and then it all ends in our waste products. And everything in between has to do with what does your body need now? Um, what does your body going to use? What are you feeding it? Are you in an anabolic state, which basically just means you're building? Are you in a catabolic state, which basically means you're breaking down? And don't confuse building and breaking down with growth, because they are not connected. This is the level of your fuel, your resources, is what we're talking about here with respect to anabolic and catabolic. Because when you're in an anabolic state, you're really in what we refer to as a fed state. And then when you're in a catabolic state, you're in the fasting state. And as everybody knows, children are thought to grow during the night when they're sleeping, so they're not in a fed state, they're in a fasting state, and they're breaking things down. But they're not breaking down their proteins and things, they're breaking down fuel sources. And so it's important for children to eat properly so that when they do go to sleep and when they do start to grow and expand their bodies, that they have the right resources in order to do so. You have, of course, in here our fuels, and we also have what we talked about before, our vitamins and minerals, as well as water. Water, everybody knows we need. And then we're going to have biosynthetic pathways. These are going to build things that your cells need. We have housekeeping genes. Those are genes that are necessary for normal cell function. So when we go through all of our metabolic processes, a lot of those enzymes are our housekeeping genes. Glycolysis enzymes, you always need to go through glycolysis, so those are housekeeping genes. And so just keep that in mind as we go through this. We also are going to store fuels. We always take in too much, so we store them so that we use them when we go on an overnight fast, for example. We are going to have to detox our waste products. Especially in a first or a developed country, there's a lot of protein available. That leaves a lot of nitrogen available that you need to excrete. And so we're going to have ways to do that. There are also, every time you have a potential for a radical oxygen species to form, you have to have a way to neutralize that radical oxygen species. And so all of this is all tied together. All our cells need energy regardless of how metabolically active they are or inactive they are. They all are going to have a sort of a net energy usage for that particular cell type. So in general, oxidation of fuels, we're going to have our fed state, which is when we are actually eating. And then this is going to lead to storage. So this leads to storage in the form of glycogen, fat, as well as building proteins, which are storing, in essence, carbon. Carbon is going to be the source of fuel for our bodies. We don't always need to use all of our fed carbons, what we're eating in our diet, and so that's why these fuel storages are important. And then we have a period of fasting where we're not taking stuff in. And so this is the case that we're going to start breaking down these storage components. We're going to start going through oxidative processes to do this. And ultimately, the goal is to generate energy, of course. ATP is our energy that we're going to need. It's going to be important for many chemical reactions in the body. It's going to help us get over that threshold that needed to move chemical reactions forward. When you're generating ATP, so you have production via oxidation of carbohydrate, lipids, and proteins, you're always going to generate some heat, you're going to release carbon dioxide. And then when you're utilizing energy, you're going to be building something, biosynthesis. or you're going to be detoxifying, or you're going to be doing some work, etc. And so those you'll be consuming ATP and generating ADP. And so this is a constant circle that we all go through as we are going through everyday life. So what do we eat? So this is what we eat. We eat hamburgers, salad, pizza, gluten-free chocolate cake. We can have curry chicken, hummus, sushi. Our bodies, what does our body see? Regardless of what we ate prior, our body only sees things as carbohydrates, proteins, and lipids. And that's pretty much it. Our carbohydrates can only be absorbed in certain forms. We can only absorb galactose, fructose, glucose. All of the carbohydrates have to be broken down into those monosaccharides. Our fatty acids are going to be taken up. Our amino acids are going to be taken up. And it depends on different types. We're going to have single amino acids and peptides, short peptides, that can be taken up. From these sources, we want to generate energy. This is the ultimate goal. And so they're all going to converge on certain points. Regardless if you're taking up glucose or fatty acids or amino acids, all of them have the same carbon skeleton that will eventually become acetyl-CoA, which will eventually enter the TCA cycle, which will eventually become ATP. So just as a side note, we just memorize these figures here, these numbers. Carbohydrates will typically give you 4 kilocalories per gram. Fat, of course, gives you more, 9. Proteins give you about the same as carbohydrates, and alcohol can give you 7, so an intermediate between the 4 of them. So what is a carbohydrate? Carbohydrates are simply molecules of sugar. This is a glucose molecule that's attached to another glucose molecule, attached to another glucose molecule, attached to another glucose, attached to another glucose, etc. And all of these are going to be broken down to a single monosaccharide of glucose. This is an example of starch, which we eat. So our potatoes, etc., will have these glucose molecules linked together. This is also the form that our body uses to store glucose. So it's called glycogen when it's stored in our body. And eventually they'll all be broken down to glucose, because glucose is really what you need to feed into your glycolysis in order to generate ATP. Now, just a few little things to point out here. We have bonds, so these red little bonds here. When you are linking a glucose molecule to another glucose molecule and they're side by side, this is referred to as an alpha 1 for linkage. So when you number the bonds in any glucose molecule, you always find the 0 or the oxygen, and then the carbon to the right of the oxygen is the carbon 1. The carbon 1 equals the anomeric carbon. And this is where interactions with other glucose molecules can occur at that anomeric carbon. The carbon 2 is here. We have carbon 3, 4, 5, and this one that's sort of up in the air is carbon 6. So glucose is a 6-carbon molecule. So it's a carbon, let me write this different. It's carbon 6 molecule. And so you're basically, when you have side-by-side linkage, it's going to be an alpha 1,4. So again, this is the 1, 2, 3, 4 carbon. So that's where the 4 comes in. And this is the 1, 2, 3, 4. And so it's that 1 carbon, called the alpha 1, to the 4 carbon. When you're doing a link that is a branch, this is going to be still the alpha 1 carbon, but now it's binding to the 6 carbon. So it's an alpha 1,6 linkage. And so those are just how you term those links. So in a glycogen or a typical starch made up of glucose, you have alpha 1,4 bonds and alpha 1,6 bonds, which are those branching points. Okay, so Carbohydrates can come in different forms. We have here shown disaccharides. So you have a molecule of sucrose and you have a molecule of lactose. And you can see that the molecule of sucrose is going to be made up of one glucose unit and one fructose unit. Lactose is made up of galactose and glucose. So again, all of these can be absorbed. So what can your body absorb? With respect to carbohydrates, it can absorb 1, glucose, 2, galactose, and 3, fructose. Your body cannot absorb sucrose. Your body cannot absorb lactose. So there has to be a mechanism for your body to break things down. And there is, and we will discuss those mechanisms. So and here's your monosaccharides. And again, here we have glucose and we have fructose. And these monosaccharides your body can absorb. Okay, so one thing to keep in mind is that there is cellulose. Cellulose is commonly found in plants. Cellulose has a 1,4 link. So you can see here, let's count these again. We have 1, 2, 3, 4, 5, 6. We have 1, 2, 3, 4, 5, 6. So we do have this 1 and we do have this carbon 4 that are linked together. However, the bond is different. You can see that the bond is going up out of the plane of the carbon 1 instead of down, and this is referred to as a beta linkage. So this is important for plants because plants need to have a rigorous cell wall, so you don't have the ability to coil. And the chains are very good at forming fibers, which again is very important for the integrity of a plant cell. Now, the crisscross pattern is for these fibers gives it many strengths, but it cannot be digested by animals. Therefore, it serves as a form of dietary fiber. It helps to regulate bowel movement. There are normal flora bacteria that can digest this. And so we're not talking about normal flora, but everybody knows that gut microbiota is important for health. And having fiber, having plant fiber in your diet is important to keep those bacteria that can metabolize that fiber in your gut, so your gut doesn't get colonized by other bacteria. So big point here is that we cannot metabolize cellulose, even though they do contain glucose, because the glucose are linked by those beta 1,4 linkages. Okay, so carbohydrates, are they necessary to eat? Of course, yes. Now your liver can make glucose, and this is a process called gluconeogenesis. But in general, if you're an active individual, it would take too much energy for your liver to keep up with the amount of glucose you would need. So what about low-carb diets? Low-carb diets, you need to be high in fat. And that's because, just as I showed you, it's going to contain the carbon skeleton necessary to compensate for the amount of glucose that you're losing from not eating the carbohydrates. So the fat can be used by the liver to make glucose. And what about fiber? Yes. So fiber is going to not be digestible except for your microflora. Okay, so now let's move into proteins. So proteins are going to have peptide bonds. And so here is where your nitrogen comes in. Remember, your body is made up of carbon, nitrogen, oxygen, and hydrogen. So the nitrogen is going to be in proteins. And an amino acid is simply a molecule that has a carboxyl group and an amino group. And these carboxyl groups and amino groups are going to join together to create peptide bonds. So amino acids join to form proteins, and the protein linkages are called peptide bonds. Now, consumption of excess protein, here's some things to keep in mind. There's no physiologic advantage. So it's not like if you eat more proteins, your muscles are going to gain more mass. That is not true. You're eating more proteins, basically you're eating more carbon skeletons attached to nitrogen. And so your nitrogen is what's going to have to be dealt with at the level of biochemistry, at the level of metabolism. And so excess protein is going to be eliminated in urinary nitrogen. And this is also going to cause an increase of urinary calcium. So you're going to lose some calcium if you have too much nitrogen. And therefore, this increases the risk of osteoporosis as well as some kidney diseases. Now, there is a protein sparing effect of carbohydrates. So dietary protein is going to be influenced by the amount of carbohydrates in the diet. So if you have a carb intake of less than 130 grams per day, then you're going to have to take in substantial amounts of protein in order to provide the precursors necessary to make glucose. Now for proteins, we know we have essential and non-essential amino acids. So with respect to our dietary requirements of our 20 essential amino acids, don't want that. We are going to need some amount of protein. So typically for men, they'll need 60 grams of protein and women 50 grams of protein. However, it depends on what type of protein you're eating. If you're eating animal origin protein, that's called high quality protein, and they contain all essential amino acids. If you are not eating those high quality animal origin, you're eating plants because you're a vegetarian, you have to make sure that you have enough input from different plant sources to have all the essential amino acids, which in the case of adults, there are 9 essential amino acids. 20 amino acids, 9 of those 20 are going to be essential, and they're listed here. You have your branched chain amino acids, lysine, isoleucine, leucine. You have threonine, valine, tryptophan, phenylalanine, methionine, and histidine. Now there are some conditionally essential amino acids. So arginine and histidine are going to be important for pregnant women and for children because of growth and need. Tyrosine and cysteine can become conditionally essential because they're dependent on one of the essential amino acids. So tyrosine depends on phenylalanine. So if you have a phenylalanine deficiency, tyrosine can become essential. And cysteine is necessary using the sulfur from methionine, which is an essential amino acid. So this just shows some of those reactions. So you can see here that you have cysteine, which is going to be dependent on methionine for its sulfur group. And then here you have phenylalanine, which can be broken down to form tyrosine. However, if phenylalanine is deficient, then tyrosine becomes essential. All right, so just a little bit about nitrogen balance. Nitrogen balance is associated with the amino acids, and the amino acids are in constant turnover. And so we have free amino acids, and they can make proteins. Remember, transfer RNA has an anticodon for the amino acid to build to make proteins. They are also precursors for essential nitrogen-containing compounds. And those would be like DNA, RNA, heme. And they can also be oxidized for energy. And then the nitrogen will go to urea for excretion. So nitrogen balance basically means that you're a normal, healthy adult. Your dietary nitrogen equals your excreted nitrogen. There are some cases where positive nitrogen balance occurs, such as in childhood and in pregnancy where there is growth. So the amount of dietary exceeds the amount of excreted. There are also some conditions where you have negative nitrogen balances, and these are typically associated with some sort of disease, whether it's metabolic or some other disease. So this is the case where you have your dietary nitrogen is less than what's excreted. And so this can be a deficiency, catabolic stress, or any of these kind of conditions. Now next up we have fats. And here is showing a fat. So a fat is essentially a carbon backbone, which in this case is glycerol. You have carbon 1, you have carbon 2, and you have carbon 3. And attached to each carbon is a different fat. So here's fat 1, here's fat 2, and here's fat 3. And so this carbon backbone with 3 fats is called a triacylglycerol. Tri for 3 fats. Now a triacylglycerol can be broken down into the backbone, which is glycerol, those 3 carbons, and the 3 fats that were attached. So here we have palmitate, the 16 carbon, we have oleate, and we have stearate. And those 3 fats can be released. So a fatty acid, let's see, a tag, which equals triacylglycerol, is glycerol plus 3 fatty acids. Now for the fat, if you break a fat down, complete oxidization, you're going to get 9 kilocalories per gram. All right, so fats are going to be stored. So let me put some of these. This is stored form. And there are a few properties that make tags very good for storage. One, it contains more calories than carbs and proteins. So tap into them when needed. 2, contains little water. There's a reason why your cells have only so much glycogen, which are sugar molecules linked together for storage, because they contain a lot of water. Fat do not contain a lot of water. Here's an example. Adipose tissue or fat tissue has about 15% water, whereas muscle has 80% water. So by having fat stored in adipose tissue, you can store a lot more than if you stored it in other tissues. Now, essential fatty acids, the definition of essential fatty acids means we cannot synthesize it. We do not have the enzymes to synthesize essential fatty acids. Why they're important is they're going to be a precursor of our eicosanoids. And these are going to be hormone-like molecules, our prostaglandins, thromboxane, our leukotrienes, platelet, not platelet, clotting cascades are going to use our eicosanoids, for example. The immune system is going to use eicosanoids, for example. And so essential fatty acid, again, we cannot synthesize. If we can synthesize it, it is not considered essential. So our essential fatty acids are two. We have alpha linoleic acid, LA, and alpha linolenic acid, ALA. We cannot synthesize either of those. Now, there are other fatty acids that people take as supplements, for example, fish oils. And with these fatty acids they're trying to get at are our eicosapentaenoic acid, EPA, and our docosahexaenoic acid, or DHA. But keep in mind, EPA and DHA, we have the enzyme to convert ALA to both of these, so they are essentially not essential fatty acids. If you do not eat any plants, vegetables, fruits, things like that, then yes, of course, you're going to have to take an EPA and DHA from some other source. But the likelihood of nobody having any plant source is very low. So DHA and EPA are not necessarily essential. All right, so here's our glycogen molecule we already talked about. Here's our alpha 1,6 bond. Here's our alpha 1,4 bond. And glycogen is made up of glucose subunits. Now, glycogen, you're going to have stores in almost all tissues, but you're going to have more in the liver and in muscle. But they're going to be different. The liver glycogen stores is going to be used to maintain blood glucose. The muscle glycogen stores is going to be used for energy. So keep that in mind. The liver is not really going to type into glycogen for its own energy purposes. It's going to type in glycogen and release the individual glucose molecules to maintain blood glucose levels. Your muscle is different. Your muscle is going to keep glycogen for its own use. Almost all cells will have some glycogen store. However, most of it's going to be found in the liver and in muscle tissue. Now, proteins is going to be used for muscles, for movement. They are also our enzymes, so they're going to catalyze all of our reactions. Proteins also form structures. They're going to form receptors, and they can be fuel sources. All right, so just a little bit about some basic metabolism terms, and again, these are sort of in flux. You typically will not see a board question on these simply because there's so many different ways to interpret these. But it's important to understand that these are still out there and people are still interested in them. So we have our resting energy expenditure. What this is just basically your housekeeping genes, the proteins that are required to make your cells function. And so this is going to be about 60%. And then you're going to have a thermic effect of food breakdown, and this is about 10%. And then our overall physical activity is about 30% on average. So these three energy requiring processes, use of their energy to generate whatever we need in order to function daily, is our resting metabolic rate, or RMR. And those three things are going to be influenced by resting metabolic weight, as well as our different in food intake, as well as our activity. So the number of calories by these in a 24-hour period is going to be our total energy expenditure. So again, if you exercise more, it's going to be the physical activity is going to be more. That percentage is going to go up. If you eat more, etc., that percentage is going to go up. So body mass index has been used by a long time to understand healthy body weight, and it's just an estimate for ideal body weight. It's typically children, it's a good, this isn't showing children, of course, but the way to measure the growth of a child is pretty well established. Body mass index for adults is kind of under flux, and it's under constant evaluation of whether or not it's being estimated properly. However, overweight is defined as about 20% above your ideal weight. Underweight is about 18.5%. And so healthy is somewhere in between that. And obese is that you're going to be greater than 30%. And so this is, you can just look at your height. If you're 6'2", and you weigh 175 pounds, then you just go over here, down here, and you're in between the 18 to 25, so you're in the healthy range, for example. So here's just a picture of our widening girth. So this is a 1985 map of the United States looking at body mass indexes in people that are adults, obviously, and you can see that we're in the blues and whites. And so, well, white means no data, so we'll ignore the white. But we're in the blue, so typically pretty healthy across the board. Go ahead of about 15, 20 years almost. Yeah, about 20 years. And you can see that now we're in the red. So this is the widening girth of America. We also have a series of attempts by the government to teach us how to eat healthy. So we have the old pyramid, which is shown here, and the idea is that you're supposed to eat more of what's down on the bottom of the pyramid and eat less of what's on top of the pyramid. And this was the idea that fats are bad, and this sort of drove the whole idea of you want a low-fat diet. And all these products came out that were low-fat, but the problem with them, they were high in sugar. And so what does your body do when it has too much sugar? It creates fat. So your body is able to make its own fat from sugar. So eating too much sugar, having a low-fat diet, isn't necessarily the healthiest way to go. We also had another pyramid that evolved, but I mean, look at this. How do you even make sense of it? It looks very confusing. It's very hard to read. So this one was replaced eventually by what is currently used is this lovely little plate. So the plate is here to show you that you're supposed to have half of your diet of fruits and vegetables. So that's pretty straightforward instead of all those little funny pictures. And then you're supposed to have more grain than protein. And then you're supposed to have some sort of calcium input, dairy products, etc. So this is the newest version. So of course, we had some cartoonists that made fun of this new plate. And they said that this is the American. We just take the plate and say, oh, I'm just taking the protein portion and everything else can go. And so in general, that is just the introduction to our different fuels and to our resting metabolic rate as well as body mass index. And so just keep in mind that diets, you have to provide all the nutrients essential amino acids, essential fatty acids, necessary vitamins, and necessary minerals. And so keep that all in mind as you try to develop your own eating habits.